Okay, so the last time we met, this was the introductory lecture for general ecology, and now we're heading into the second lecture for the semester. And I wanted to start, first of all, welcome back. And I wanted to start with this iconic slide again, just because it really it impacted a lot of people's lives. It impacted the science of ecology. It actually impacted science in America. A lot of money was delivered into the science curriculum as a result of actually of Sputnik, but this also was really intriguing. A lot of people my age look at this and they do remember it. They do recall, and I certainly benefited from all the scientific education that I received because the U.S. really wanted to compete at that level. And it had, you know, as I joked last time, it went from Tang to IBM computers to everything else. We really gained a lot. And yet it was only just to go to the moon. So it seems really dumb. It's like basic research. Why do you care? What's the application? Humongous. Those applications come. You don't know what they are yet. Anyway, so that's where I wanted to start today. And I will kind of go give you a quick overview of the lectures from the last time. And from lecture 1, recall that an ecologist collects and analyzes data. An ecologist, or ecology, spans scales from the minute to the global. And I had that cute little graphic that showed that the minute is knowable, usually. Global is really hard, hugely relevant, but difficult to track all the competing factors. And if anything, ecology is loaded with competing factors that are impacting the system you're trying to study. And I didn't have this in the last slide, but I throw it in here because it's important to realize that ecology, of course, informs our efforts in conservation and restoration ecology. Our campus, EFB, is very interested in conservation as well as restoration, and increasingly so. Sustainability as well, right? You can't understand how to keep a system sustainable without understanding the system. So again, back to basic ecology. Basic information, you don't know the impacts until you get there. And then, boom, this is really applicable. All right, and very quickly, I also went over this idea about the current view. Nature is not static. Variation is the rule. And even rates of change vary. So as an example, our DNA, we have sections of DNA that are constantly changing with each generation, and others that have remained stable since humans came along, or since bacteria. And so variation, even within our DNA, it's not as stable as you might imagine. Parts of it are evolving very rapidly. Okay, and then all species evolve, some fast, some slow. Again, the rate of change varies across organisms. And with the current view also, humans are definitely part of the system. And we are part of nature. We are animals. A great example is that we are invasive species. Boy, you ever want to see the impact of a species moving into an environment on that system, watch a human migration event. Yeah, we're kind of done filling in the spaces on the planet. But you look at Hawaii when the Polynesians arrived, and then look at Hawaii when the Europeans arrived. We lost a lot of species that were very unique to that ecosystem and could not withstand that invasive species pressure from humans. Okay. So now we get into today's lecture. Karl Popper, 1968, note the date. There was a lot going on back then. He comes along. People at the time, I think it's fairly safe to say, were very good natural historians, very good at observing nature, but they weren't very good at testing their hypotheses. And Popper wrote a paper that said ecological studies that do not explicitly test their hypotheses do not advance the science. So you can even generate the hypothesis, but if you don't do anything to test it, to see if it's valid, that's just words on a piece of paper or coming out of your mouth. So he says that's a problem. And there was a little bit of physics envy going on back then, where ecologists and biologists didn't have what physicists had. Physicists could really test their ideas because the system, especially the small ones, with mathematical models, they were really much more advanced. And Popper was suggesting we should start doing this with ecology as well. It avoids incorrect conclusions and avoids bias of the observer, which is inescapable, and poor observations, which is something that can be corrected by the observer, but it happens. What can you do? So he brings in the need for statistics. It's not my fault, it's Karl Popper's. We'll get back to that in a little bit. To do a good experiment, you have to have a good experimental design. And you need controls, although that is a lot easier in a laboratory setting. Kind of hard when you're looking at Mount St. Helens as a volcanic eruption. What were the impacts of that? It's an N of 1. We don't have a lot of replication there. We don't have any controls. So in ecology, sometimes you can do very good experimental design because you can kind of, I'll show you this in a little bit, but you can basically manipulate within the system and come up with replications and controls, but sometimes you can't. Global warming is a good example. There is only one Earth right now that we have to study global warming or climate change. Replication is important. I put greater than 3, greater than and equal to 3. That's a bare minimum. But I do have a paper that I published that was my replication, and the statistics weren't very powerful because there wasn't a lot of replication, but it was such an obvious pattern that the statistics did support the rejection of the null and the acceptance of the alternative that XYZ was happening. It is important with replication to avoid pseudo-replication. So you want to take your samples, let's say you're doing something with amphibians in ponds, samples from multiple ponds, not multiple samples from one pond, because those multiple samples from one pond are more similar to each other just by design, just by definition, than they would be to those coming from a separate pond. And so you want to take your samples from multiple ponds to get the variation of the habitat. Even though they're all ponds, each one has its unique features, and that will impact your study. So you want to avoid this thing called pseudo-replication. Yes, you're replicating, but it's all within one system. It's not as powerful as multiple zones, multiple locations. Okay, more good experimental design. Random sampling is always very important. This is another interesting topic because some would argue you can never have a random number. When I generate random numbers for my sampling, I plug in, well, I have my computer, calculator, probably can't see it, but it's there, calculator or my cell phone, and I've got a little feature on there that generates a random number. It starts with some seed, therefore it's not random. But it's okay, right? You could also not even do random. You could just do systematic sampling every 10 centimeters or 10 meters or something like that. That's okay. You just have to do something to avoid bias. And it's best to come up with some version of a random sampling scheme. Large sample sizes, very important. In this class, because we have so many students, we can get large sample sizes for our reports because we have 10 labs and 300 and some, 200 and some students. Also, there is a difference between descriptive versus manipulative studies. Descriptive would be going out into a field and watching a robin turn its head and then poke its beak into the ground and pull out an earthworm. And you say, this robin is listening to the sounds underground. He hears the movement of the worm, pokes his beak down there and pulls it out and has some food for itself or its young. That's kind of pre-Karl Popper. What would be more powerful, and is more powerful, is if you could figure out a way to manipulate the system, maybe figure out what sound, record the sound, and then play it back out in the field. See if a robin goes and tries to get an earthworm. Something as trivial as that, or more, elaborate. But you have to have a way to manipulate the system to see if the ecology is following your presumptive idea about what's happening there. Okay, so you manipulate and then measure the response. This image is from your textbook, and it's an image that these researchers were interested in defoliation, and they couldn't figure out what was going on. They put a netting around this plant that was getting, should have been getting hit hard by insects, but wasn't in this system. So they put this netting around it that allowed the insect in, but it didn't allow birds. And you can't see it, probably. Let's see if I can get this to work. So here is a little flag. I don't know if you can see my cursor. There's a little flag, and here is the stick that's left right now. And I'm going to advance, and this orange line is going to trace. It's a little off. But there are no leaves. What happened? Through that manipulation of keeping birds out, those birds weren't preying on the insect that was defoliating the plant. So that's a very nice manipulation and pretty easy data. Of course, you'd want to replicate this wherever you found the plant and get solid information out of that. But very nice manipulative study. Okay, we always start with an observation. So this is on top of Mount Tamalpais, just north of Golden Gate, the Golden Gate, or the Golden Gate Bridge. And it's where I was a young sapling. I was a master's student at San Francisco State, and my professor was walking around these areas and coming up with tons of interesting projects. And he saw this pattern and was reading some papers that were coming out of Oregon to suggest that Douglas fir, I'm going to do something real quick here. Click on that, and I can't see where it says laser. Is it there? Well, we'll just go with this. Okay, this is easier to see. These are Douglas fir trees right there and here. So in the 50s, Army training went wrong. The two planes collided above this area. One of them went into the Pacific Ocean. These were Corsairs. The other one landed pretty close to where this picture was taken. There was a big crater right there in my research plots. Note that, and then a big fire. This is in the 50s. Big fire swept through the entire area. This is California. Everything has been reestablishing since. Note that the Douglas fir only establishes in this light vegetation here, but not out here. That was his observation. And then the Oregon folks were saying these plants are associated with fungi that won't support Doug fir. These plants are associated with fungi that Doug fir can actually use for its nutrient acquisition, for getting nutrients out of the soil as well as water. So it looked like there was something going on here, and if you draw a line right there, you could set up this nice experimental design to say there is a difference between, for Douglas fir, between landing here and landing here. And so for my master's, I set about trying to test every hypothesis I could think of. Why is there that difference? In this very tight little area, it's not a different amount of rain. The heat should be the same other than underneath the vegetation, etc. Okay, so I'll kind of work through this. And the hypothesis, the null, I'll come back to this again in a bit, but would be written as the difference in Douglas fir survival in Arctostaphylos, back in this shrub, and Adenostoma out here is no greater than expected by chance. But I gave the talks like this back in the day, early 90s, and a very effective, very advanced mycologist, senior mycologist came up afterward and said, that picture you showed says it all. Clearly there's a difference. Okay, enter caulopoper. Let's figure out why. Enter me. Yes, good place to grow or land if you're dug for seed. By the way, the wind blows from the Pacific Ocean out here across this area. So I didn't test wind for seed dispersal, but it's well known. There were dug fir behind me. They're all over. And they've all established within the last 50 years after that fire. And they're not establishing in these patches, the adenostoma. So the first thing I did is I went out and I planted Douglas fir seed underneath those two vegetation types. Where they were establishing in Arctostaphylos. This is manzanita. You might know Arctostaphylos uva ursi is all over the place, all over the world in the northern hemisphere. In California, it's a species-rich area because that's the diversity center. So anyway, that's where the Douglas fir were establishing, and this adenostoma is a rosaceous plant in the Rosaceae that there was no establishment. So I planted underneath these shrubs. They're about 1 meter to 2 meters tall. There's one of my little Douglas fir seedlings in this little figure right there. And then this is the Arctostaphylos sticks coming up, again, going up to about 2 meters. I recorded their germination first, and I start with 100% of those seedlings that germinated, and then went out repeatedly and recorded the survival of germinants in my plots. Most die, as you can see, they're dying very quickly from April through basically the summer drought. This white area is the summertime, things dry up. It's miserable for a seedling if you haven't figured out a way to get water. So plants in the Arctostaphylos are dying off as the soil's dry. Plants in the adenostoma are dying off, but quicker and more severe. And eventually they go to 0 in September in the adenostoma. But those in the Arctostaphylos actually make it through the summer drought. Not many, but in ecology you only need a couple, right, to survive and go on to the next generation, hopefully. And in fact, those seedlings, by and large, did survive because now the rains had come in this second gray box, and they survived until I went out there and picked them, in part curious to see what the roots look like, and in part because I had done a manipulation and I should remove that gene influx. I wanted to get that stuff out of there because that wasn't part of the original system. Even though it was Douglas fir, those seed had been collected somewhere else in California, and so I didn't want to bring that new gene pool in. So they were eventually picked. Okay. Here's an interesting side point that I want to use to express another little angle on sampling design. So here I'm collecting the data on survival in this first portion of the graph, right, or the whole thing. I'm going out once a week to all my plots and collecting those data. And then note that I don't really do a lot of collection right in here, which is too bad because a lot was going on. But there was an ESA conference right here, Ecological Society of America, and I wasn't able to get back out there for a couple of reasons until September 11th. Different September 11th than you're thinking. And so I wish I had gone out more frequently. Did I need to go out as many times in the early portion? Probably not. By the time we get down here in September, I'm only going out every month or less, and that was enough. Pattern was captured. So this is another figure from your textbook, I believe. I have to go back and check that. Sometimes they pulled it on some editions. But the point here is this is the variable, and let's just say it's 100% of whatever you're looking at. And this is the time, let's say, or distance where you're measuring that particular thing. In my case over here is measuring seedling survival. If you go out every day, you're wasting a lot. You're capturing the pattern perfectly. It's this really nice kind of sine wave. You're capturing that very nicely if you go out every day, but what a waste of time. If you only go out every 5 days, I believe it is, you don't capture it. You get this line, this middle line with the big blue dots. That doesn't capture the pattern very well at all, except maybe here. So the trick is to say, okay, I think I'll go out every 5 days where the squares are. And that does as good a job as going out every day. And you save yourself a lot of time. So pilot studies are a very useful thing. For those of you who might think of going on into graduate school, I can't emphasize enough the importance of collecting a little bit of data to inform how you design your experiment. I may emphasize it enough for you in this class, but I think I'll just leave it here for now. Pilot studies are very helpful so you can design your experiment for your sanity as well as capturing the ecology the best. Okay, back to my study site. So the question is, why is Douglas fir only establishing, or why is it establishing better, pretty much only in the Arctostaphylos in this system, not in the Adenostoma? And so the next phase of my master's was to figure out what might be driving that. By the way, that seedling study was a manipulative study, right? I went out and planted seed, followed the germinants through the summer drought. That's a manipulation. Now I'm going to do a bunch of observation studies where I want to see what are the differences between these two habitats, that is Arctostaphylos and Adenostoma. And for these studies, there were no seedlings or sapling, no Douglas fir around. I found other patches that were in close proximity where I did, a measurement, series of measurements out here, and then a series of measurements in the more positive Arctostaphylos zones or patches. And here are those data. It's a little bit complicated, but I will show you. I measured light, so I took a light meter underneath on the soil in the Adenostoma, and I reject the null. There is a difference. In fact, Adenostoma is like rosemary. It's little tiny leaves, and tons of light come through the Adenostoma. So there was plenty of light coming down here for a Douglas fir to get established, which tends to be a light-loving conifer. Soil nitrogen. Adenostoma, again, there was a difference, but there was more nitrogen in the soil of Adenostoma than in the Arctostaphylos. Because Adenostoma, I can show you this in a second here, right here, this flowering plant here is a different species. It's Ceanothus cuneatus, and it has nitrogen-fixing nodules. So turns out there's quite a bit of nitrogen in those soils. So again, reject, but it's in the wrong direction, right? Doesn't fit the observed pattern. Soil P was kind of all over the place, incredibly variable. I have to accept that, okay? But there didn't seem to be a difference, is the bottom line. So it's not better for phosphorus in the Arctostaphylos stands where the Douglas fir was establishing. Same for potassium and calcium and magnesium. So those are sort of your fertilizer nutrients that you would get on any fertilizer you buy off the shelf at a garden store. Water was better under Arctostaphylos. Because it has big flat leaves and they lay down a litter layer, they actually held in the water. And reject the null hypothesis, and it is in the right direction. It's better under Arctostaphylos for a little Douglas fir to get established. And then, of course, the mutualistic fungi. When I went out and started collecting mushrooms, I never saw any in Adenostoma, okay? And I did find them in Arctostaphylos. Whether they got onto the roots of the seedlings, I didn't know at this time. That would come later. But these two things fit the pattern. And so I reject a number of simple hypotheses, and I'm left to accept the possibility that it was soil moisture or mutualistic fungi or both that were contributing to the establishment pattern I had observed. And that might be interesting to you. You might say, wait, 2 possible explanations? Is that okay? And at my defense, I had a graduate student who, I don't know, I must have pissed him off at some point, but he would not let me go at my defense. As professors, my parents were there. He's like, you can't, why couldn't you rule out whether it was water or mushrooms and blah, blah, blah. And I was like, well, you know, mushrooms need water. Why can't you have two? And I eventually passed anyway, despite his best efforts. No, sorry, I'm not, I have some PTSD from that event. But no, this is not a problem in science, right? You can have two explanations that are perfectly sound, and you can't sort them out. You can set up conditions where it looks like light is a wave. You could set up the conditions to make it seem like light is a particle. So this is kind of I found this on the web and I never did find out who wrote it. So I apologize. If you're out there, please contact me and I will gladly give you a full acknowledgement. But if you look at this, not only does it say wave, and this is a W, but it says particle. Particle. So, and then there's the wave also, and a particle. So anyway, you can have dualities like that. What you do is you set up your hypotheses, your experiments, and give it your best shot. In the end, you want to come up with your best explanation. And over time, you or someone else can try to sort that out better. And so, you know, case in point, Earth is the center of the universe. No, it's the center of the solar system. No, the sun is the center of the universe. No, actually, okay, each time another group came along or somebody came along and provided evidence, and the smart scientists said, okay, that we need to modify what we thought. And science is, if anything, a constantly changing body of knowledge that we learn from new evidence. And I've known plenty of people, including myself, who have a hard time rejecting an early hypothesis. But it's very important to be able to do so to advance the science. Okay, so that's it for this segment. I'm now going to move in next segment to some statistical methods and explain how much you really need to know for my class. This isn't statistics. This is ecology, but ecology uses statistics. So I'm going to give you some sort of theoretical background so that you can understand what you are reading, and what you're collecting, the data you're collecting, and how to analyze it from the theoretical point of view. I'm not going to be requiring you to understand how the math is informing that view, but you do need to know how to use the math to get to the answers. So I want to provide that information for you. So yeah, we'll see you next time, and have a good day. Cheers.